All right, so today we're diving deep into the world of proteins. You know those amazing 3D models that computers can spit out? They look incredible, right? But here's the thing. How do we actually know if they're real? How can we be sure we're looking at a scientific breakthrough and not just, well, a really pretty piece of digital art? That's what we're going to figure out. Think of it like a detective story. So yeah, this is the big question scientists have to ask every single time. Every new protein model is basically a suspect in a lineup. It might look totally perfect on the surface, but just one hidden flaw could send researchers on a wild goose chase for years. So, how do they put on their detective hats and tell the difference between a genuine masterpiece and a masterful fake? Okay, so our investigation has to start with a little bit of healthy skepticism. We've got to ask why. Why can't we just trust these computer models right out of the box? Well, it all boils down to what a protein actually does. You know, a great way to think about it is like a key. A protein has this super specific, really complex 3D shape, and that shape is everything. It's what lets it lock into other molecules and kick off all the chemical reactions that keep us alive. But if you get that shape wrong, even by a little bit, the key doesn't fit the lock, and suddenly that model is totally useless for figuring out diseases or designing new medicines. The stakes, yeah, they're massive. And that really gets to the heart of the problem. There's this great quote from Dr. Etheosala that just nails it. We can't always believe on programs. They often mislead or sometimes may be wrong. And that's it, right there. The software that builds these models is, don't get me wrong, absolutely amazing. But it's not perfect. It can get thrown off by bad data, or it might make a guess that just doesn't work in the messy reality of biology. And this brings up a really, really important difference. See, a computer is just doing math. It can build a model where all the numbers work out perfectly, the angles are right, the energy scores look great. But just because it works on paper or on a screen doesn't mean it's actually possible in the real world. A scientist has to be the one to step in and be the reality check. They have to ask, okay, but does this thing actually exist in a living cell? Can it do its job? That's the huge gap between a simple calculation and biological truth. All right, so we know things can go wrong, but what are we actually looking for? What do these errors look like? It's time to profile the usual suspects. In our little detective story, these are the common bad guys that can totally mess up a protein model. So the very first clue a scientist looks for is something called sequence identity. Just think of it like checking a fingerprint. See, these models are usually built by comparing them to a known protein, which acts as a template. Sequence identity just measures how similar the new protein is to that template. And there's this kind of rule of thumb. If the similarity is over 30%, you're probably on the right track to getting a decent model. But if it drops below 30%, whoa, the chance of major errors goes way, way up. It's a super important clue for sure, but it's definitely not a guarantee. And that brings us to the most wanted list for protein modeling errors. You can have some real classic mistakes here. Number one, you could just pick the wrong blueprint to start with. That's an incorrect template. Or maybe the blueprint is right, but you line things up all wrong. That's a misalignment. Sometimes there are parts with no blueprint at all, so you have to guess, and those guesses can be wrong. You can also get weird distortions and shifts, but the most common one of all? Getting the tiny little side chains packed together incorrectly. It's a classic. So, we know who the suspects are, but knowing who to look for is one thing. Actually catching them is the fun part. It's time to open up the Investigator's Toolkit and check out the high-tech forensic gear scientists use to hunt these errors down. Okay, so this whole category of tools is called Model Quality Assessment Programs, or MQAPFs for short. And you should really think of them as the detective's complete forensic kit. This isn't just one magnifying glass. Oh no. It's a whole collection of different programs, and each one is designed to basically stress test the model from every possible angle, just trying to find a weak spot. And if there's one central hub for all this, it's a server called SAFES. You can think of it as the validation headquarters. It's this one-stop shop that brings all these different forensic tools together. A scientist can just upload their suspect model, and SAFES will run it through this whole battery of tests and spit out a detailed report card on how good or bad it really is. So what are these tools actually looking for when they're running their tests? Well, pretty much everything. They double-check the original alignment. They look for what are called stereochemical irregularities, which is just a fancy way of asking, hey, are these atoms arranged in a way that's even physically possible, or is this just a jumbled mess? They look for bonds that are stretched or strained. And the big one, they flag any arrangement of atoms that's basically never ever seen in the natural world. Okay, let's zoom in a little bit. how does one of these tools actually uncover the hard evidence? How do they catch an error red-handed? To get this, we first have to talk about something called rotamers. So, quick science refresher. Proteins are made of amino acids, and each amino acid has this little arm that sticks out, called a side chain. Now, this arm can move, but it doesn't just flap around all over the place. It actually prefers to settle into a few very specific, very stable positions. These are the rotamers. You can think of them as the side chain's most natural or comfortable poses. So, if a computer model shows a side chain twisted into some bizarre, unnatural shape, that is a massive red flag. And this is where a specific tool from that save server comes in. It's called ProCheck. And honestly, the best way to think about ProCheck is like it's a lie detector, but for the model's physical shape. It knows exactly what a real, stable protein is supposed to look like, and its whole job is to see if your model is telling the truth. And one of the main ways it does this is by creating something called a Ramachandran plot. Now, we don't need to get lost in the details here, but the important thing to know is this plot is basically a map that shows which angles in the protein's backbone are good and which are bad. If any part of the protein shows up in what's called a disallowed region on that map, well, that's the equivalent of the lie detector needle flying off the charts. It's the smoking gun. It's hard evidence that something is seriously wrong with that piece of the model. So, after putting the model through the ringer, running all these checks, looking at all the evidence from every angle, once all those red flags have been cleared, our protein detective can finally reach a verdict. And this is the whole point of the exercise. A model that makes it through all of that isn't just a good guess anymore. It's not a prediction. It becomes a valid piece of evidence. It's a trustworthy blueprint that scientists can actually build upon for real-world research. Case closed. But why does this matter so much? I mean, what's the big deal? Well, with a model you can actually trust, you can do amazing things. You can design a new medicine that fits perfectly into a protein's little pocket to stop a disease. You can finally understand why a tiny genetic mutation causes a protein to fold wrong. And, maybe most practically, it saves scientists from wasting years of work and millions of dollars chasing dead ends based on a model that was a fake all along. So the next time you see one of those absolutely stunning 3D images of a protein, just remember there's a whole detective story behind it, a rigorous investigation to make sure it's the real deal. And it really leaves you with a big question, doesn't it? If a protein's shape is its destiny, what kind of future can we build? What kinds of cures can we discover if we make absolutely sure we get that shape right every single time?